Okay, this is going to be the last lecture of our adventure this summer together. To be honest, I'm going to skip, well, not skip, but kind of fly through the slides. I've tried to break out some of this, what I'm referring to as alphabet soup. And so some of the slides are wordy, and I'm not going to waste your time reading those to you. I've really tried to pull it out and hit high points and also throw away some of the complexities that you may have noticed when you were working through this chapter. So it's actually really exciting. I've always loved signaling, but in getting back into it, I realized that it is a complex beast. So like I said, what I'm going to try to do is focus on some of the big pictures. I'm not going to spend a lot of time getting into, again, the alphabet soup. Now, why messenger molecules are cool to me is because they help coordinate and control important cellular functions. And so there's lots of different ways that they do this. We can have endocrine cells that drop molecules into the blood directly. Those molecules are then sent throughout the entire body, diffusing out of the bloodstream and finding receptors on target cells. We can have molecules that we've talked about before, like I mentioned with prostaglandins, that are localized hormones that act very near the site that they're released. But in most of these cases, and in fact, I believe all of them that we're going to talk about today, we're going to talk about receptor-mediated messaging. And so again, the big picture idea is, well, we need to get a message from outside the cell to inside the cell so the cell can respond and do something. So that is very important to understand that general idea. And so this is, again, one of those slides that I'm not going to waste time reading to you, but just take some time to read through this. So one of the reasons I was so late in getting all this together was I really tried to take some time and take what the book was saying and really put it into concise terms. And again, kind of bullet point those terms for you. So again, to save time and my voice, I'm going to skip through a lot of these wordy ones. And like in this case here, there's lots and lots of information on here. Now, you're not expected to know this. So just understand that we have a bunch of different endocrine glands, and they release a bunch of different types of hormones that do a bunch of different types of things. Now, you may say that's really general, but that's about the level that we need to understand. If we really wanted to get into this, we would just get into a physiology class. But again, this being biochemistry, we want to look at the methods that these signals get from the outside of a cell to the inside of a cell. And so biochemical signaling, typically we get a hormone that binds a receptor, and I've got a little picture over there on the right. And so you can kind of follow that. There are some cases where the hormones don't bind the receptor, and there's some steroid and some thyroid hormones actually cross the the cell membrane and actually the nuclear membrane and bind in the interior of the nucleus and do their action there. And so that would kind of make sense for the steroid hormones. If you remember their general structure, very similar to cholesterol, so they should be able to passively diffuse across the cell membrane. Now, a lot of charged hormones, and there's several types. So some of these hormones that are charged are amino acid derivatives. Some are actually polypeptides that can range from a few amino acids to several hundred amino acids. Insulin is a great example of that. And then you've got these steroids and lipid-based ones. And so again, you can see a class of the type of hormones down here. You can see where they regulate. Again, these that are regulating in the nucleus tend to cross the membrane and get right to the nucleus and do their job there. These right here are plasmine membrane receptors, or they may be second messengers that we'll talk about. And like I mentioned, the prostaglandins or these eicosanoids, they tend to be localized hormones, where some of these others, peptides and these catecholamines that we'll talk about, are endocrine and released into your blood vessel and delivered throughout your body. So again, we're going to kind of focus on the hormones binding the receptors. The receptor will transfer the signal from the outside of the cell to the inside, and then you get some type of intracellular response. And we'll talk a little bit about kinases, and I think we might even get to phosphatases. And we really won't get into this idea of enzyme cascades that amplify the signals. I talked briefly about that when I talked about blood clotting. But one of the things is if a hormone binds to the outside and is transferred to the inside, and you get a second messenger, or you turn on this inside process of the second messenger, you can take one signal and all of a sudden it can get amplified dramatically from one binding on the outside to multiple signals. And they may be the same signal, just multiplied, or actually different signals. Again, we'll touch on a little bit of that. So here's just some more classes of the hormones. I think it's interesting to look at their structures. Right here, you should be able to look at that and say, oh, that is a hormone that is derived from tryptophan. So you can see kind of the general tryptophan. This is actually melatonin, regulates your sleep-wake cycle. This is a steroid hormone, so this is going to be one of those that can actually cross over the membrane and do its action in the interior of the cell and the nucleus. And then these are going to be the peptide hormones that need to bind the outside of the cell and get their signal transferred to the interior. And again, just a list for you. And again, since I'm skipping over these, you're not expected to know that in detail. Again, a very wordy slide here. What's interesting about this is if we look at pancreas, it is a glandular organ, so lots of glands. And so it's got this exocrine gland, and that is dedicated to producing digestive enzymes. And so exocrine glands tend to dump their signals or proteins into a duct. Endocrine tend to dump their signals or molecules into blood vessels. So that's the difference between an exocrine and endocrine. But in the pancreas, you find both of them. So we talked previously about trypsin, chymotrypsin, alpha amylase, phospholipase A2. And so again, these are secreted by the duct to the small intestine where they can help digest food. Now, a small amount of the pancreatic tissue consists of what's known as the islets of Langerhans. And so these are endocrine glands that, again, they dump their the hormones into blood vessels. And so these islet cells actually contain three types of cells: alpha cells, beta, and delta cells. And alpha cells secrete glucagon, which is a peptide hormone. And this is secreted in response to low blood glucose. And so what happens is this will go and cause the breakdown of glycogen. Insulin is secreted in response to high blood glucose levels. So go bind to cells to get the cells to take up the glucose and get it out of the blood. Somatostatin is also secreted by the hypothalamus, but also by these delta cells, and it inhibits the release of insulin and glucagon from the islet cells. So it's released from a separate place and actually works to balance out these glucagon and insulin. Could you imagine you don't want your pancreas just to dump a bunch of glucagon and insulin into your blood at the same time. So you need to delicately balance those two together. So that is the pancreas. We also have adrenal glands, and they consist of two types of tissue, the medulla and the cortex. So you got the adrenal medulla and the adrenal cortex. So the medulla is really an extension of your sympathetic nerve system, autonomic nervous system. And then you got the adrenal cortex, and from that, you release two catecholamines. And I've shown you pictures of what a catechol is and what generally a catecholamine is. And so those two hormones that come from the adrenal medulla, the adrenal medulla, I think I missaid that before, but we're talking about the adrenal medulla. Those are norepinephrine and epinephrine, or noradrenaline and adrenaline. And you can see in the structure below. So again, that is the adrenal medulla. Now the adrenal cortex releases your steroid hormones, your androgens and estrogens. I'm just going to let you read through this because you actually have a couple of different types of adrenergic receptors. So these are going to be the receptors that actually receive the signals from these hormones that come from the adrenal glands. So right now we've just talked about norepinephrine and epinephrine. We're going to talk about some more of these adrenal hormones, but you got norepinephrine and epinephrine, and they can bind to these receptors on cells, these beta adrenergic receptors and alpha adrenergic receptors. And essentially these mediate the fight or flight. So at the very bottom there, you can see the effects are directed towards a common end, mobilization of energy resources and getting those energy resources to where they're needed so the body can act. And I'll let you read through those, the roles that they play. So you may have heard of beta adrenergic and alpha adrenergic receptors before. Now here's something that you need to know, the definition of an agonist. That is a substance that binds to a receptor and evokes a response. So it is a doer. Now an antagonist, you've probably heard that word before, it's someone that antagonizes. But in biochemistry, an antagonist is a substance that binds a receptor and fails to elicit response. So essentially it blocks an agonist action. So it antagonizes the enzyme or the receptor, and it inhibits that action. So you got agonists that cause action, antagonists that inhibit it. Now again, like I mentioned, you don't need to know the specifics of the alpha adrenergic receptors and the beta adrenergic receptors, but there's some interesting things there that these different adrenergic receptors, like I said, that are targeted by epinephrine and norepinephrine, depending on the molecule, can be blocked. So we got phentolamine, and it is an antagonist for alpha adrenergic receptors. And then you got isoproterenol, and that stimulates beta adrenergic receptors. And then you got propranolol, and it's got no effect on alpha adrenergic receptors, but it blocks the beta adrenergic receptors. So no effect on the alpha, but it blocks, and so it's used to treat high blood pressure and attacks. So again, this latter half you don't really need to know, but these are highly targeted receptors, these alpha adrenergic and beta adrenergic receptors, and so you can target them differently, and they do different things. So also in the adrenal hormones, you have, like I said, the different tissue, the adrenal cortex, and you've got three different types of hormones released. And we talked about these back in the lipids. In fact, this is the exact same slide from the lipids, and so I won't read through this again, but essentially you've got the glucocorticoids that affect metabolism, again, energy release. You've got the mineral corticoids, and just like the name says, mineral, they're going to regulate water and salt. And then you got the gonatocorticoids, and they affect sexual development and function. And you've got the male specific and the female androgens and the estrogen progesterones. Okay, so let's talk a little bit about binding to these receptors. And just for an example, you've got growth hormone, and it's 191 residue polypeptide. It is produced by the anterior lobe of the pituitary gland. Again, details that we're not necessarily going to need to know, but just how this process works is the growth hormone binds the growth hormone binding protein. Oh, these guys are great. So GH binds GHBP. And this is where we get into the alphabet soup. So yeah, some of these things make sense. Others, you're not quite sure. So this growth hormone binding protein forms a dimer that then binds the growth hormone, again, on the outside of the cell. And this will cause changes on the interior. So in this picture here, we actually have HGH, which you may have heard of as human growth hormone, and then the dimer of the human growth hormone binding protein. So again, out of this section, the general take-home message is that this signal needs to get from the outside to the inside, so you can explain why only certain cells respond to certain hormones, because they have the receptors for those hormones. And list some hormones produced by the pancreas. Well, we talked about those in the adrenal medulla, and we talked about those in the adrenal cortex. And specifically, what type of molecules are these? And those are based on the steroid hormones. Don't really worry about symbolizing the biological effects of insulin, glucagon, norepinephrine, androgens, estrogens, and growth hormones. But in general, understand that things like insulin, glucagon, and norepinephrine, these are involved in metabolism, energy release. Androgens and estrogen are more in development, sexual development, and growth hormones, of course, in growth. And so definitely know the definition of an agonist and an antagonist. Okay. So we're going to talk about a specific type of receptor known as the tyrosine kinases. And so these are referred to as the PTKs, protein tyrosine kinases. So again, the alphabet soup, but protein tyrosine kinases, what they do is they will take a protein, and on that tyrosine, they will add a phosphate. And that is what a kinase does, is it phosphorylates something. So like I said, we're going to talk about it first, receptor tyrosine kinases, heterotrimeric G proteins, and just a little bit about the phospholinositide cascade. So these are three systems for transducing signals. And again, transducing, we're moving the signal from outside the cell to inside. And so in protein tyrosine kinases, what we're going to do is on a protein, find a tyrosine residue and add a phosphate. And that phosphate comes from ATP. So this happens, well, let me go back. So many of the membrane receptors are of this type. And what they have is a extracellular domain. So that's outside the cell. You have a region that is buried within the membrane, and then you have a region that is inside the membrane there. We'll see a better schematic of that in a second. And so what happens is you get a hormone to bind to the outside. In fact, this one is an insulin binding. So insulin actually binds here. And what's going to happen is on the inside of the cell, these domains are actually going to come over and autophosphorylate each other. So they're going to add a phosphate group to themselves when a hormone binds to the outside. And so it's, so then that change allows the binding, and then it actually phosphorylates a specific target protein. So these get phosphorylated when they bind the receptor, and upon phosphorylating them, they turn into kinases and will phosphorylate other targets. So again, this general idea, we get a hormone that binds a receptor. The receptor transfuses the signal to the inside. You get an intracellular response. And that intracellular response may involve a second messenger or a kinase that goes and adds phosphates to things, or phosphatases that actually remove phosphates from substrates. And again, you may get the enzyme cascade. So an example, so you got insulin here, and it's going to interact with the cell by a receptor tyrosine kinase, an RTK. And when insulin binds, it is going to initiate a cascade of events that leads to increased glucose uptake and metabolism. And that makes sense. So if you have a large amount of glucose in your blood, you release insulin, and that causes this cascade, so the cells will uptake that glucose. So if you can't make insulin, or your cells don't recognize insulin, that leads to the disease diabetes. So this receptor tyrosine kinase, this RTK, it's unusual for being a dimer in the unliganded state. And what that means is a lot of these receptors will form a dimer once the receptor comes in, then they come and they form a dimer. This one here actually is a dimer that is kept together by these disulfide linkages. So this is an insulin receptor. It's a receptor tyrosine kinase. So it receives something, and then you've got these tyrosine kinases on here. And so when the ligand binds, it induces these dimers to actually move closer together. It brings these cytoplasmic domains, which are these protein tyrosine kinases. So again, this alphabet soup, you've got RTKs, which is this whole thing. This is a receptor tyrosine kinase. But when you go and bind that receptor, it brings these domains here that are tyrosine kinases. And so these are called the protein tyrosine kinase domains, the PTKs. It brings them close together, and they cross-phosphorylate each other. So when they get close to each other, they start adding phosphates to each other. And then that phosphorylation actually activates these guys so that they can phosphorylate other things. So kind of interesting. They phosphorylate each other, that turns them on so they can actually phosphorylate other molecules. Okay, so this is looking at the cytosolic. This is the tyrosine kinase domain. So let me go back. This is just looking at this portion of this receptor tyrosine kinase. And you can imagine if we're going to autophosphorylate, we need to be able to bind ATP so that we can add phosphates to our tyrosines. And so this is a crystal structure, and what they did is they're using an inhibitor, and instead of an oxygen here, you put a nitrogen. And that makes this so that you can't transfer this phosphate group. So it binds as an inhibitor and allows you to crystallize, and then this domain doesn't turn over the ATP. And so you can see this is the nitrogen of that inhibitor. And it looks like in this one, they already have phosphates attached to several tyrosines in here. So these are the tyrosines that got phosphorylated. This is the ATP analog that binds to phosphorylate another substrate. So again, somewhat confusing, but let me walk through that one more time. These guys phosphorylate each other. So we're going to add phosphates to tyrosines there. So let me go to the next slide. You can see those phosphates here. And when those phosphates are attached here, this can now act as a tyrosine kinase. So what it can do is take tyrosines and add phosphates to them. So if it's going to add phosphates to something else, it needs to be able to bind ATP. And so there's the ATP that is bound that can be transferred to some substrate. Okay, so here is a diagram of what happens when you get this autophosphorylation. So you have what are referred to these activation loops, and you can see 3 tyrosines there. They're in the ball and stick model in the inactive form on the left. And so when that receptor binds on the outside, these guys are going to autophosphorylate themselves. So one domain is going to come over and put phosphates on the other one. And when you put phosphates on that activation loop, that causes this activation loop to change its conformation, and you get a pretty wide swing that actually opens up the binding site for other target proteins. So you phosphorylated this protein, you made it active, so now it can phosphorylate other proteins. So there you go. Again, one of these slides I'm not going to read through to you, and I'm going to leave the discussion there. If you read in the book, they talk a lot more about certain things like these insulin receptor substrates that are actually the ones that get phosphorylated. These insulin receptor substrates are proteins that contain these SH2 modules. These SH2 modules actually stand for Sark homology 2 domains. And so what's really interesting is some of this makes sense. Insulin receptor substrate. So this is a substrate that's going to get phosphorylated. But in this insulin receptor substrate, you have these SH2 parts that have this really weird name that really doesn't describe what they do. So this is where we get into this alphabet soup, and we'll just stop. So hopefully you understand that insulin binds. We activate these domains that can then cause phosphorylation of other groups. And so we've moved the signal from the outside to an enzyme on the inside that can do something. And if you want all the details, you can look at this diagram here and just look at all those fun acronyms there. So like I said, we're low on time and mental energy, so we're just going to appreciate this. But again, this key that the signal from insulin has moved to the inside, and now we can actually phosphorylate something. So you can see here specifically it's this insulin receptor substrate that gets phosphorylated and causes the cascade. And again, you can see what this does. And eventually what it does is it makes it so that you get more of these GLUT1 type receptors that get glucose to come up into the cells, as well as other things that happen. So anyway, alphabet soup. I was supposed to hide this slide, but at some point I was going to flesh it out. So yeah, anyway, just imagine that this was hidden, but I was just trying to take every single one of their acronyms there and put it into a Rosetta Stone so we could translate it. I got tired. I gave up. So, in fact, that was just where I was going to leave that discussion. I think I was going to hide those two. Sorry. Now, I did want to do another one of these cascades. It's similar to the insulin, but this is the protein growth factor, and this is referred to as a RAS signaling cascade. And again, look at how similar it is. In this case, these domains aren't cross-linked. So what happens is when this protein growth factor binds, these two subunits come together. And again, when they come together, they phosphorylate. Now, it's a little bit different here. When we phosphorylate, that phosphate on this receptor actually recruits these domain proteins that are connected to other proteins that recruit other proteins that recruit other proteins that cause a signal to occur. So this is a little bit different than the tyrosine kinase, because what's happening here is we're actually recruiting proteins, and by doing that, that allows something else to occur, which are these G proteins. Okay, now these are monomeric G proteins, and I'll just briefly talk about them. That in this form, RAS is inactive when GDP is bound. When this binds to this SOS protein, it encourages the binding of GTP. And so when GTP is here, it's on. When it's GDP, it's off. And this GTP causes this RAS protein to do a bunch of stuff. And again, if you wanted to know, you could walk through. So there's a description of steps 1 through 3, 4 through 6, and 7 through 10. Again, just understand that this general idea of these proteins coming together when it binds a hormone on the outside causes changes on the interior that could either, for the tyrosine kinase, cause it to activate as a protein, or to recruit other proteins that then get turned on. So these receptors can act as an enzyme or recruit other proteins and turn them on. Okay, so that is receptor tyrosine kinases. So let's jump in and just briefly talk about heterotrimeric G proteins. Now, right there at the end, I just talked about some G proteins. Those are monomeric. These are heterotrimeric, meaning I've got 3 subunits that are all different. And so, again, this is one of those places that I tried to condense a lot of that very thick information. But I'll let you read that, and let's just jump into talking about this. So you've got GPCRs, which are G protein coupled receptors. So this is the protein that is going to bind a hormone from the outside and convey it to the, not convey the hormone, but convey the signal to the inside. And what it's going to do is it's going to convey that signal to this trimeric G protein. There's 3 of these G proteins, an alpha, beta, and gamma subunit. And so in this case here, it's got GDP and it is off. It is bound to the receptor and just waiting for something to happen. Well, when this signal binds the receptor, what it does is it causes GDP to be released from the alpha subunit, also release of the beta and gamma subunits, and binding of GTP. And so now you get the and the G alpha subunit dissociates as well. Sorry, I should have mentioned that. So when this binds, all 3 of these subunits dissociate from the receptor, and in doing so, GTP binds the alpha subunit. And then that alpha subunit, now that it's turned on, it will go and activate something else. So I'm going to scoot back a little bit to show you the similarities to what we just talked about here. In this case, this is a monomeric protein with GDP that's off. You put GTP on it, and it is now active and will cause other enzymes to be active. But we're not doing this through this G protein coupled receptor. But I want to make the connection between these because I think it starts to make more sense in how our body does this. And this isn't such a different idea. You have a G protein that when it binds GTP, it's turned on. Now, how that G protein gets the GTP is different here. That's really the only difference. And what it is you just have a receptor that interacts specifically with these G proteins, where in the previous example, it acted indirectly. The receptor binds, causes dissociation, the GDP comes off, the GTP comes on, and this is now active and will activate another protein. Okay, So hopefully that's clear, because what we want to do is focus on this activating adenylate cyclase. And so adenylate cyclase actually is a very important enzyme that is what's referred to as producing a second messenger, which is cyclic AMP. So here's another picture of it. In this case, we're going to have epinephrine bind to the outside. That is going to cause this trimeric, this heterotrimeric protein to dissociate. All three of those subunits are going to dissociate. And as they do so, the alpha subunit is going to bind GTP. And now this is active. Notice it's glowing halo around it. It's active. It bumps into adenylate cyclase, and adenylate cyclase is now active. It should have a glowing halo around it too, woo, since it's turned on. And what it does is it turns ATP into cyclic AMP, which is referred to as a second messenger. Well, where's my first messenger? That's epinephrine. Your second messenger is cyclic AMP, and that cyclic AMP is now a messenger that acts within the cell. So it's actually like an interior hormone, but it's going to activate protein kinase A. And protein kinase A goes from pink, or in this case red, which is inactive, to the active form. Okay, so you're going to see this diagram because we're going to watch through each one of those cells. So here is the G protein coupled receptor, the GPCR, and I'm just showing you the part that is buried within the membrane. And so actually the receptor site is actually tucked in within the interior a little bit. So this right here is a high affinity agonist. So I'm going to give you a second to figure out if it's going to turn this on or turn this off. So this is a beta adrenergic receptor. It binds this high affinity agonist. Well, an agonist turns it on. So it is going to cause this to transfer that signal to the interior. So essentially it's an epinephrine analog. So, and here's its structure. And so when this binds, it's going to cause some conformational changes, and that is going to cause, like I said, the dissociation of the G proteins that are coupled to this receptor. So again, this alphabet soup makes sense, G protein coupled receptor. So let's go to the next step, the heterotrimeric G proteins. I've identified those in a box there. So they consist of alpha, beta, and gamma subunits. Let's take a little bit closer look at them. So that's this region down here, and you can see the parts that are labeled alpha, beta, and gamma. And these are G proteins, and the reason, again, they're G proteins is because they will bind GTP. And so in this case here, it is actually the structure, in this case, it's unbound with GTP, or sorry, GDP. And in this case, the GDP is bound to it. And so you get a conformational change with the GDP bound. Okay, so again, some more information that I put there. So if you read through this, hopefully this will make sense. But again, details that would just complicate the picture at this point, because we want to move on to the final step of this, and that is activation of the adenylate cyclase. So again, we're kind of breaking it down into 3 points: binding to the receptor, the heterotrimeric G proteins, and then activation of the adenylate cyclase. So adenylate cyclase, let's actually see what it does. So referred to as AC, and it cyclizes ATP. And so how it does this is you have a free 3' hydroxy group, and it will come in, attack that phosphate, kicking off a pyrophosphate, an excellent leaving group, and makes this cyclic structure. And you can see that this is cyclic adenosine monophosphate. So it's not AMP, it's cyclic AMP. And this cyclic AMP is going to act as a second messenger that transmits the signal. that originated outside the cell, and the cyclic AMP is now going to do the work inside the cell. So let's talk a little bit about adenylate cyclase to give you a picture of it. Now, cyclic AMP is polar. It freely diffuses as a second messenger. So you can see why we wouldn't be able to transfer it from the outside to the inside. And epinephrine is polar. So epinephrine essentially won't get across that membrane, but we want it to signal and get inside. So really what we have is epinephrine comes here to the outside, and essentially that signal gets transduced into cyclic AMP. And again, the enzyme that does that is adenylate cyclase, and so you can see adenylate cyclase here. Okay, and finally, the last step there, adenylate cyclase, cyclic AMP activating protein kinase A. So we'll just talk a little bit about that. So let's take a closer look at protein kinase A. And protein kinase, hopefully you're picking up that a kinase is an enzyme that phosphorylates other molecules. So we're seeing that that is a common way to turn on and turn off proteins. So what we're doing in this case is making a enzyme that's floating around the cytoplasm and turn on so it can phosphorylate other targets. Now with the protein receptor tyrosine kinases, those are embedded in the membrane. Now this case, the protein kinase can actually float around and phosphorylate different targets. And so it's going to phosphorylate at the residues that have OHs, serine or threonine. Now the proteins that protein kinase phosphorylates has a sequence, an arginine, arginine X, where X is any small residue, and then it has a serine or a threonine. That's where we're going to add the phosphate, and then it's followed up by a hydrophobic residue. And so in the blue and purple here is protein kinase A. You can see that it is in complex with ATP. And you can see the sequence right here of the target sequence. Actually, sorry, this actually looks, anyway, it says that it's bound with it looks like it's actually bound with ADP. It looks like there's two phosphates, and it looks like it's already transferred the phosphate there. So yeah, I took that out of the book, but it looks like this is ATP that's already transferred the phosphate to this white part. Oh no, actually, sorry, it can't. I need to back it up. I'm confused. I did take this from the book, and you're probably confused, so let's back up. If I had time, I'd edit that out. So let's just start. Say, hey, protein kinase A is blue and purple. And we've got ATP bound here. To be honest, I don't know what that is. I'll go back and look. But here in orange is its target. So cyclic AMP has turned on this protein kinase A. Protein kinase A is a bound ATP, and it's found its target sequence, which again is this arginine, arginine, small residue, serine or threonine, and then a hydrophobic residue. Now this is an inhibitor. What they did is they replaced alanine at that position. So there is no hydroxy group that can get phosphorylated in there. And so anyway, that's what protein kinase A does. Now the targets of protein kinase include enzymes involved in glycogen metabolism. So epinephrine binds the cell. Well, epinephrine, if you remember from the very start, is important for the release of and the mobilization of energy resources. Well, glycogen is our glucose energy storage within the cell. So epinephrine binds to the outside. It causes the heterotrimeric G couple, or the G protein, to dissociate. The alpha subunit binds the GTP. And that goes and activates the adenylate cyclase. The adenylate cyclase then activates, or sorry, makes cyclic AMP. Cyclic AMP then activates protein kinase A. And then protein kinase A goes and adds phosphates to proteins involved in glycogen metabolism, and we release glucose. So this essentially is involved in the fight or flight response. So ultimately, what it does is it activates glycogen phosphorylase, which makes glucose 6-phosphate, and that is what's used in glycolysis. And we're not going to get much into metabolism. Actually, we're not going to get into metabolism at all. So there you go. There is an example of this. And if you want to see this in all the glory, there it is. Yay! So there is a lot of complexity here. You can look at this in your book and they will talk about it. I didn't break out all the steps, but let's just run through this one more time in generality, and we will show you just how these signals. So you have some stimulatory external signal that binds this G protein coupled receptor. This G protein coupled receptor, you've got alpha, gamma, and beta subunits, and they dissociate. And what happens is you will get GTP to bind. Now, what cholera toxin does, that's very interesting. I'd read that little box in there on toxins. In fact, we'll talk a little bit about that later, but essentially inhibits the hydrolysis of this GTP. So this is now active and will come over to adenylate cyclase. And so if cholera toxin is around, essentially this alpha subunit with GTP is just on and just keeps adenylate cyclase on constantly. And you just keep producing cyclic AMP. We'll come back to that. But again, this is now on with the GTP, turns adenylate cyclase on, producing cyclic AMP. 4 of those will bind to the phosphokinase A. Phosphokinase A will take ATP and add some protein that's inactive and turn it into an active protein that then goes and does a cellular response. So again, you look at this from afar and it's just overwhelming, but hopefully you could walk through that process there. Now, something that we really don't have time to talk about, but the same type of thing can be inhibitory. So you can actually get G protein coupled receptors that are actually inhibitory. So you get an inhibitory sequence that now has a different alpha. In fact, this is referred to as the I alpha. And so you may have wondered what the S over there is for, and that's the stimulatory, and this is the inhibitory alpha. So these are going to be different proteins here. Notice how they actually have different shapes there. And so they can bind to a different receptor and the same process, but what happens is when they bind GTP, they're on. And what I mean by on is they're active, but the effect they have is inhibitory. And so when you look at the signaling cascades, lines that have this line across there, essentially an elongated T, that means inhibit, and arrows mean activate. So you can see these are all arrows here. Cholera toxin inhibits the hydrolysis of GTP. Perasus toxin inhibits this here. So essentially, when you have the perasus toxin, this is going to be is going to prevent the hydrolysis of GTP. So then this can't bind GTP. And so then you never get the inhibition of this. Okay, so again, this inhibits the hydrolysis in this case. Okay, so there's drugs that affect cell signaling. And so some cases, caffeine, well, sorry, actually, let me walk back up. These are purine derivatives, and they act as stimulants because they antagonize adenosine receptors that act through this inhibitory process. So essentially what happens here is these antagonize and shut down the inhibitory process. So I'm going to give you a second to try to figure out what that's going to do. Essentially what it's doing is it's shutting down this pathway here. So you get caffeine to bind here, and it binds here, it antagonizes this and shuts down this. It does. It works through the inhibitory, but it antagonizes here. So it prevents things from binding and turning off. So what is that going to do to your cyclic AMP? So I kind of butchered it. Hopefully from the diagram you picked it up. So these methylated purine derivatives are stimulants. They antagonize the inhibitory G proteins. And so here's just some example. But essentially the antagonism results in an increase in intracellular cyclic AMP. So really by antagonizing this site here, this isn't happening, so we're not shutting off our adenylate cyclase. So in this process here, what we're seeing, this is an agonist. This is causing this to occur, which turns off adenylate cyclase. If we antagonize it, then none of this is occurring, so we're not turning off adenylate cyclase. And you just keep producing cyclic AMP, which is going to cause you to release more glucose, which is going to help stimulate you. Essentially, you're kind of bypassing the epinephrine and you're giving yourself a low dose of adrenaline. And so caffeine and adrenaline work in similar processes by activating the interior of the cell to release metabolic molecules and open those processes up. So in this case here, this is cholera toxin, and I briefly talked about it. And so what it does is it actually binds to the heterotrimeric protein at an arginine. So the cholera toxin actually causes NAD to bind covalently here. And so now adenylate cyclase is now always active and produces way too much cyclic AMP. Now this isn't as bad as caffeine, but if you keep your cyclic AMP on, essentially at some point you're going to over exercise your cells and burn up all your ATP and release all your glucose, and ultimately you'll die. Similar things would happen if you take too much caffeine. But okay, so there is another one. So this is phosphodiesterase, and this limits second messenger activity. So in any chemical-based signaling system, the signal molecule must eventually be eliminated. You want that cyclic AMP to be turned off. So in the case of cyclic AMP, the second messenger is hydrolyzed to AMP by enzymes called cyclic AMP phosphodiesterases. So diesterases, they're going to cleave these phosphate bonds. So these phosphodiesterases are inhibited by a series of drugs. So essentially, they also stop the breakdown of cyclic AMP. The previous examples we talked about upped the concentration of cyclic AMP. Essentially, this is going to inhibit the removal of cyclic AMP. So it's not that you're going to have more of it, you're just going to remove it more slowly. And so these are inhibited by a variety of drugs involved in disorders such as asthma, congestive heart failure, depression, erectile dysfunction, inflammation, retinal degeneration. And so this is known as Viagra. It is used to treat erectile dysfunction. It inhibits PDE5, so this phosphodiesterase, which hydrolyzes cyclic GMP. So essentially, it increases the amount of cyclic GMP that you have, We won't get into the full process of what happens there, but it involves nitric oxide, NO molecule, increase in blood vessel dilation and absorption of blood into nether regions, and fixes the problem. So these phosphodieesterases are involved, again, in shutting down these second messengers, but if you target them, you can also treat diseases by increasing the amount of these second messengers. Okay, so those are heterotrimeric G proteins. And just briefly, we're going to talk about the phosphoinositide pathway. We're really just going to just briefly talk about this. So phosphoinositide pathway, it mediates the effects of a variety of hormones. And this pathway requires a phosphorylated glycerophospholipid. And this glycerophospholipid is a minor component of the plasma membrane's inner leaflet, so it's found on the inside. We need a G protein coupled receptor. We need a heterotrimeric G protein and a specific kinase. And so we're going to mix all that together. But in this case, we're not going to activate a protein kinase C. We're going to release inositol 1,4,5-triphosphate, IP3. We're also going to involve the production of calcium and also 1,2-diacylglycerol, or DAG. So phospholipase C reaction. So here is your phosphatidylinositol 4,5-bisphosphate. This is found on the inner leaflet, and you can see that over here. Okay, so you're going to get some signal from the outside. And that signal from the outside is going to activate phospholipase C. And when you activate phospholipase C, remember phospholipases cleave the bonds. So phospholipase A1 cleaves here, A2 cleaves here. Phospholipase C is going to cleave here. And so when you activate phospholipase C, it is going to release this molecule from the inside, and it is actually going to go bind a receptor on the endoplasmic reticulum, and that causes the release of calcium. Okay, like I said, we're not going to get too much into the details here. Just understand that we can release other second messengers besides cyclic AMP. In fact, calcium 2+ is a second messenger. This IP3 is a second messenger, as well as that diacylglycerol. And so here is the legend for, this process right here. So similar to what we saw before, we have an external signal that binds to the G protein coupled receptor. We get the dissociation of these subunits. The alpha one here binds the GTP, and it activates the phospholipase C here. So instead of activating the adenylate cyclase, we activate phospholipase C that again cleaves that bond and releases this inositol triphosphate here. And when you release that inositol triphosphate, it's a second messenger. Again, it can go bind the endoplasmic reticulum, cause calcium to be released. Calcium can then go and turn on some kinases that are inactive, and when it turns on these kinases here, these kinases then go and activate other proteins. So a different way of turning on the kinase, instead of turning it on by binding cyclic AMP, it's being turned on by binding calcium. What's interesting is the leftover molecule, the diacylglycerol here, that you cleave that IP3 from, is now a second messenger, and it can bind to protein kinase C. And protein kinase C that can then take a protein that is inactive and actually cause it to be phosphorylated. Or sorry, yeah. Actually, let's go back and read 7, sorry. Yeah, the nonpolar DAG remains associated with the membrane where it activates protein kinase C and to phosphorylate and then by modulate the activities of a number of cellular proteins. So essentially the protein, this protein kinase C here will take an inactive protein and activate it. There you go. So you're expected to replicate this on the exam. Now I know only 2 or 3 people will listen to this lecture. So if you say, I refuse to replicate the last slide on the exam, you'll get +1. That lets me know that you made it through the last lecture. And so you may say, why in the heck? Actually, that's probably not the word that you used. Why in the heck are you showing this? Okay, I just wanted to show this to you because the signaling cascades can get very, very complicated. And if you look at just insulin binding there are multiple things that are happening, a cascade of reactions that are happening. So from binding just one receptor, you get tons of response from the inside. So a very small amount of insulin can cause a large amount of cellular activity. And again, +1 for emailing and say that you're not going to replicate this on the exam. All I do is want you to understand in general how we can get those signals from the outside in and understand in general, again, the 3 ones that we talked about. The protein tyrosine kinase, the heterotrimeric G-coupled proteins, and I'm butchering their names, but it's only 11 o'clock at night. And then finally, the phosphoinositide pathway. Oh my goodness. Let's just call it that. Let's call it a semester. Thanks. Good luck on the final. And we will hopefully talk to you soon.